Right, guys, let's have a look at biopsychology. First of all, that big essay that I gave you on endogenous pacemakers and exogenous Zeitgebers. So first of all, I chose this because this is the last year that this essay could possibly be asked, any of the rhythms essays, in fact. And this hasn't come up for a while, so it's one to watch. All of them are, to be honest, but this... yeah, I just have a feeling about it. So you need to be careful with it because the answers weren't on the whole great. What it basically is, essentially, is a glorified circadian rhythms essay. You're talking about the sleep-wake cycle, which is that circadian rhythm that you've already done an essay on a couple of essays before. So what you're essentially doing, and why I'm describing it as a glorified version, is that you're using the key terms endogenous pacemakers and exogenous Zeitgebers in order to explain how the sleep-wake cycle works. So it's basically the same essay. So the things that are missing from your AO1 are mainly the biology. So endogenous pacemakers are internal bodily factors that impact the sleep-wake cycle, and your master pacemaker is the SCN, which is a bundle of nerve fibres found in the hypothalamus of each hemisphere, that take in information about light from the environment and send that information to the pineal gland and instruct it to either stop producing melatonin if it's light, because you need to be awake, or start producing melatonin if it's dark, because you need to get to sleep. So all of those internal concepts regulate your sleep-wake cycle via the SCN. Now, there are also these things called exogenous Zeitgebers, which are external time givers, like light. So when light is received by the eyes, that process then becomes biological, becomes an internal one. So exogenous Zeitgebers, like light, noise. So it's really hard to sleep when you live in a city when there's noise constantly outside. That keeps you awake. That's an exogenous Zeitgeber. Or meal times. Meals tell us, oh, it's daytime, I need to be awake because I'm having breakfast, lunch or dinner. That's why we don't eat at night. It doesn't help us sleep. It gives us energy. It keeps us awake. So you're explaining those factors. And then in the AO3, you are linking any study or implication you make to either support of endogenous pacemakers, support of exogenous Zeitgebers, or evidence that refutes one of those. You're kind of using it to weigh up which is more important. There was some strange AO3 that was stated, like a particularly difficult one was when you talked about jet lag as AO3. In evaluation, you can combat jet lag by switching to the meal times of the new country that you move to. Don't set it up like that, because that doesn't sound like AO3. That's just pure description. And that's not evaluative. However, changing that to in evaluation, one implication of exogenous Zeitgebers is if you use them effectively, you can combat issues that humans face, such as jet lag. And it's actually, there's another really important key word that most of you missed out, which is entrainment, which is like the adjustment period that your body goes in when it does change time zone. So you can use meal times to speed up the process of entrainment and counteract and change or update your sleep-wake cycle. Seafra's Cave. Tell me what it's about, and then explain to me that it supports the idea that endogenous pacemakers are free running in the sleep-wake cycle, and they're more important than exogenous Zeitgebers. They trump them because we didn't need light in order to stay in a circadian rhythm in that study. Same with Folkard, same with Weaver. All three of those studies show that endo is more important than exo. Lots of you talked about like Apple iPhone, night shift button, shift workers, but you didn't really frame it as AO3. You just sort of described that this thing existed. Well, that's not analysis, is it? That's not evaluation. It might be stated on the PowerPoint, but you need to use your exam technique to make that evaluative. So with the night shift button, in evaluation, one implication of exogenous Zeitgebers and endogenous pacemakers on the sleep-wake cycle is that Apple has spent a fortune trying to create a red light mode on their iPhone, which doesn't impact circadian rhythms. So it changes the exogenous Zeitgeber of that into red light, so that the SCN doesn't start that process of waking you up and stopping producing melatonin. It manages the process differently in order to prevent our sleep being affected. So in that way, like, research on exogenous Zeitgebers and endogenous pacemakers has been used to improve our lives by companies like Apple, or casinos use it against us. They disrupt our sleep-wake cycle. So we normally would be going to bed, but we're staying in that casino where there's total absence of light. We don't know whether it's night or day because of all the other factors that they do, and then we lose track of the sleep-wake cycle, and we either stay in there all day in the absence of light, or we stay up all night, but we keep spending our money because that sleep-wake cycle has been so disrupted by manipulating our pacemakers and the Zeitgebers that we're exposed to. So I guess what I'm trying to say is, make sure AO3 is set up as AO3 and it doesn't just sound like a description. Every single AO3 point needs to be linked back to the question, and make sure you give me the actual biology of what endogenous pacemakers particularly are. That was poorly answered. The EZ bit was okay, but don't set up AO3 as if it's your own personal opinion. Say things like psychologists have shown, psychologists have researched, psychologists have discovered. You have to be quite careful with that. The second question in this section, question 7, which asks for a similarity and a difference between fMRI and ERP, wasn't always well answered. The number one thing to remember is that it is not temporal validity or spatial validity, it's temporal resolution and spatial resolution. And I can't give you full marks if you use the wrong terminology, because it's just completely wrong. So fMRI has amazing spatial resolution, whereas ERP has bad spatial resolution. It doesn't produce a map of the brain like the fMRI does. However, in terms of temporal resolution, it's the other way around. So ERP has incredible temporal resolution and fMRI has poor temporal resolution. fMRI produces a map of the brain, whereas ERP does not. It produces basically, like, it's deciphered from a graph, from EEG data. So fMRI measures brain activity directly via blood flow, indirectly via blood flow, whereas ERP measures brain activity directly via electrical impulses. Loads of people said things like, both of these involve the person being alive, not deceased. I can't give you marks for things like that, guys. Come on. I can't. That is scraping the barrel. What other things did people say? Oh, both are non-invasive, don't involve use of a radioactive tracer like PET does. Also, both are carried out in response to a stimulus, or they measure the brain while it's actively engaged in a task. I did give marks for they can both be used in order to detect brain abnormalities, with some examples, things like Alzheimer's. Just definitely one to look at. I thought that was quite an easy question and I was proven wrong. Then we have this question about briefly evaluate localization of function in the brain. Now, the main issue with this question is that loads of you, first of all, evaluated lateralization. So localization is the idea that a specific brain area, let's say my amygdala, through into the centre of my brain here, is responsible for a particular behaviour, emotional process. And we know it is. The amygdala is key in emotional processing. That's maybe where fear is located. That's localization. How do I know this? Well, PET scans or fMRI scans have shown that that area is active when experiencing that specific emotion. That would have been a great AO3 point. You could use that one from psychopathology. Lots of you referenced Tolving, which was great, and how Tolving found that different types of long-term memory are located in different parts of the prefrontal cortex. Brilliant. But if you just spoke generally and didn't tell me where something was localised, I couldn't give you the marks. Phineas Gage, his personality entirely transformed after his prefrontal cortex was damaged, showing that personality, or those aspects of his personality, are localised in the prefrontal cortex. Lots of you use Sperry, which you can't use because Sperry is lateralisation, not localisation. So lateralisation is different. It's the idea that one whole half of my brain is dominant for a particular function. That's not localisation. Other things you could talk about were things that went against localisation theory. So Lashley showed that learning is a holistic process. It doesn't involve just one part of the brain, but many, many, many parts of them. It's a holistic process. So you can look at the PowerPoint as well. There were lots of things that you could say here, but you needed 2 really decent studies, really, or AO3 points in order to gain the full 4 marks. Lots of people gave me AO1. Okay, that's biopsych.